and this is the C. elegans Lab Report.
The overall purpose of this experiment is to manipulate the RNAi system in order to determine the function of the specific gene we were given in C. elegans.
Before we attempted to switch off our gene in the C. elegans, we observed wild-type worms on NGM agar with OP50 bacteria for them to eat. The wild-type C. elegans were moving forwards in S-like waves fairly quickly, and they were easy to spot because they were opaque. The eggs were very small, and there was a fair abundance of them throughout the plate. As the stages progress, the adolescent stages of L1, L2, L3, and L4 have increasingly long and thick bodies, with the adults being the biggest.
After we observed the wild-type worms, we cut a 1 centimeter squared piece of agar and transferred it onto the new agar plate with NGM light agar with
ampicillin, an antibiotic that kills bacteria that is not wanted on the plate.
IPTG, a gene promoter that causes the worms to produce more mRNA of the selected gene, and RNAi bacteria with some form of double-stranded RNAi.
We then observed the worms under the microscope to check that they were still alive.
When we observed the changes in the phenotype of the worms, we found that not only were there quite a number of dead worms, but most notably, the worms that were dead were shriveled up and almost entirely transparent. What had caused this change was blistering on adult worms. Individually, the blisters looked like a batch of transparency on their otherwise opaque bodies. The blisterings were primarily prevalent in the adult stages, which is usually when the blisterings get big enough to pop, causing death. As you can see here, the dead worms appear to be quite small and could even be mistaken for an adolescent worm, but this is due to the fact that after enough blisters pop, their inner fluids leak out, making them look smaller without it. Once blisterings have completely covered their bodies, they were almost entirely transparent. This blistering also induced slower and less efficient movement, as can be observed throughout the plate.
But how did this happen?
Well,
after we transferred the C. elegans to the new feeder plate, the worms ate the RNAi bacteria with the double-stranded RNAi with a mirror copy of the gene that we were trying to silence.
The double-stranded RNA entered the worm cells, and then, because it contained the mirror copy of the gene, the RNAi molecules identified it as a viral piece of RNA, and it got chopped up into small, short pieces of RNA called small interfering RNAs.
Then, these siRNAs bonded to several RNAi proteins to form RISK, RNAi-induced silencing complex, which then proceeded to unzip all of the double-stranded siRNA, making it single-stranded RNA.
These RISK molecules then searched for any mRNA that matched the RNA that it had bonded to.
RISC eventually bonded to all of the identical mRNA and separated it, even the ones the cell itself produced, causing all of its mRNA for this gene to be destroyed, meaning that this gene is now unable to produce its protein product and has been effectively silenced.
The gene that we successfully silenced in the worms is called BLI1, and it turns out that it codes for the production of collagens that are essential to the proper formation of the cuticle, which is like an exoskeleton that not only acts as a barrier between the environment and the worm, but maintains locomotive functions in the C. elegans. Specifically, that gene encodes for a major cuticle cross-linking enzyme that, when absent, causes the two major layers of the cuticles to rub against each other, and as we previously observed, results in blister forming.
this experiment wasn't actually terrible, and it was really interesting to see how the RNAi can be used to determine the specific function of genes.
We really enjoy the ability to see the results of the gene silencing in the C. elegans in action. We also learned a lot on how to be more independent and responsible through conducting this experiment.